This video is on the 3 pathways of complement activation. The first pathway is the classical pathway. It is part of the adaptive immune response as it starts with an antigen-antibody complex. This all starts with the C1q macromolecule. C1q is a molecule made of 6 proteins composed of 3 pieces each. When combined, the structure has a stem with 6 legs. A collar fits on the stem and is made of 2 C1r and 2 C1s molecules. To start the classical pathway, the C1q molecule must interact with 2 antibodies. Once attached, the C1r in the ring is activated. This will cause the second C1r and both C1s's to activate. The activated C1s will cleave C4 and C2, starting the complement cascade leading to the cleavage of C3 and the corresponding result of inflammation and opsonization. The next pathway is the lectin pathway. This is considered part of the innate immune system since it does not need antibody interaction to be activated. Instead, it identifies and attaches to the carbohydrate surface molecules on microbes called lectins. The protein receptor of the lectin pathway is a 4-legged structure called mannose binding lectin, or MBL. This lectin recognizes and binds to the carbohydrate markers on bacteria, fungi, and even some viruses. The MBL is associated with other proteins that are threaded through the MBL structure. These other proteins are called MBL-associated serine proteases, or MASP. There are 3 MASP proteins. MASP2 closely resembles C1s of the classical pathway. Once the MBL is bonded to the pathogen marker, the MASP is activated. Once activated, MASP will start the cleavage of C2 and C4 to create C3 convertase and break down C3, leading to inflammation and opsonization. The final pathway is the alternative pathway. Since it does not require any interaction with an antibody, it is part of the innate immune system. This pathway is divided into 3 pathways. The first is the tick-over pathway. C3 is constantly being made and released into the vascular system, and at times it will be activated. This usually results in the degradation of the molecule, but when this happens near an infection, some of the C3b molecules will attach to the microbe. Both factor B and factor D are needed to complete the pathway. To stabilize C3 convertase, it must bind to properdin. Once associated with properdin, the pathway can continue to generate numerous C3b molecules in an amplification process. This pathway ends with C5 convertase, resulting in lysis with MAC and inflammation. Sometimes, the alternative pathway does not need an activated C3 to get started. In the properdin-activated pathway, properdin will attach to the microbial cell wall. This allows a base for the C3b and factor B to attach, then attracting factor D and the production of C3 convertase. However, for this pathway to work, C3b must be present from the tick-over pathway. The last pathway is the protease-activated pathway. Blood clotting factors circulate in the plasma, and such factors as thrombin can cleave C3 and C5. The mere triggering of the blood clotting cascade can activate the alternative pathway and thus the complement cascade. These multiple pathways ensure that the invading microbe will be recognized and eliminated. This concludes the video on the 3 pathways of complement activation.